name's Urvi, and my thesis deals with investigating toxicity of low concentrations of nickel in rainbow trout by examining proteomic profiles of skin, mucus, and blood plasma. Nickel is a widely used metal with a projected increase in demand since it's extensively used as a component in the manufacturing of steel and to make nickel batteries in electric and hybrid vehicles. Recently, the discovery of a nickel sulfide-rich ore in the Ontario Ring of Fire has piqued potential mining endeavors in the James Bay lowlands in northern Ontario. This raises the possibility of a local freshwater contamination due to mining activities. Adverse impacts of nickel are observed at high doses and can be witnessed at many levels of biological organization. At the molecular level, nickel can alter expression of antioxidant enzymes. Cellular level impacts include genotoxicity and reduction of cells involved in immune response, and nickel predominantly accumulates within the kidney of fish and is linked with an increase in iron content in tissues, whereas in the whole organism, nickel exposure significantly impacts respiration and reduces the fecundity of fish. An important aspect of my thesis also deals with the use of non-lethal and non-invasive tools to study toxicity in animal experiments. Traditional toxicology methods involve the sacrifice of fish to sample and study their organs, which is unfavorable and contradicts the 3 R's of animal research, namely replacement, reduction, and refinement. Non-lethal and non-invasive tools are beneficial because they can be used to study endangered species and also offers the ability to sample the fish over time, improving the strength of the study and providing a robust analysis of effects. The objectives of my study include the use of epidermal mucus as a non-lethal and non-invasive tool to identify temporal and dose-dependent proteomic responses of trout exposed to environmentally relevant concentrations of nickel, to study the dose-dependent response of the proteome of rainbow trout using blood plasma as a non-lethal tool, and to identify potential biomarkers of nickel by comparing proteomic profiles within and between mucus and blood plasma. Most studies expose fish to higher doses of nickel between 50 and 10,000 micrograms per liter, and therefore the known toxic impacts of nickel are documented within this range. However, I wanted to study the impacts of nickel at the level of the proteome, which is more sensitive to changes in the environment. Therefore, I used lower levels of nickel to study impacts on fish. Based on the ambient nickel levels at the Ontario Ring of Fire, which is low at about 0.2 micrograms per liter, the avoidance threshold, and the CCME guidelines for nickel, my 3 nickel treatments included the 2 low doses, 1.5 and 4.9 micrograms per liter, and 1 high dose of 45.7 micrograms per liter of nickel. For the waterborne exposures, rainbow trouts ranging between 250 and 400 grams were divided among the treatment groups with 2 replicate tanks containing 7 and 8 fish each, and the nickel stock solutions were delivered to the tanks using a peristaltic pump. The duration of the exposure lasted for a total of 30 days. On day 0, we tagged the fish, sampled their skin mucus, and initiated the exposures. We also sampled skin mucus of fish at the halfway point, and on last day, we sampled both skin mucus and blood after 30 days of exposure. The mucus and blood underwent proteomic analysis to identify changes in protein composition. Mucus samples were filtered and centrifuged using an Amicon ultracentrifugal filter tube to concentrate the proteins. Blood samples from the fish were spun to separate the plasma. The proteins in the plasma and mucus were digested and fragmented into peptides using formic acid, and the peptide sequences of the fragments were generated using liquid chromatography mass spectrometry. The peptides were then identified and quantified by matching their sequences to the rainbow trout reference proteome using SpectrumMill and Skyline softwares. Statistical analysis was done using lemma on R, and proteins that were significant were searched against databases to conduct gene enrichment, functional, and pathway analysis. To conduct statistical analysis, I used lemma, which is the linear model for microarray data. The analysis is commonly used for microarray, RNA-seq, and transcriptomics data, but has also been used recently to analyze data obtained using a proteomics approach. It uses an empirical Bayes approach to fit the model. I used lemma to identify proteins that were significantly affected by the main effects of nickel treatment or time, and the interaction effects of nickel treatment and time in the mucus of trout. For plasma, since samples were only collected on day 30, the proteome was analyzed only for the main effect of nickel treatment. All analysis was conducted using a Benjamini-Hochberg p-value or false discovery rate of less than 0.5. The Venn diagram shows the proteins that were impacted by nickel treatment, time, and interaction effects. The expression profiles affected by the mean and interaction effects can be shown by the following plots. The first four show some of the proteins that were affected by nickel treatment, the second set shows proteins affected by time, and the third set shows proteins affected by the interaction effect. Since the interaction effect and main effect of time encompass proteins that are affected by nickel treatment, you will notice in the protein profile plot shown here and in subsequent slides that the proteins do not show a strong dose-dependent response with nickel for the concentrations used. The model suggests that there is a stronger effect of time on the abundance of proteins. Similarly, in plasma, nickel treatment significantly affected the abundance of 1240 proteins. This number is likely higher because of the effects of time and interaction were not considered in this model. The plots on the right show the dose-dependent response of proteins in blood plasma based on their abundance. To simplify the analysis, my presentation will only cover a part of my results, which includes the analysis of the proteins that were affected by nickel treatment in both mucus and plasma. To understand the effects of nickel on the proteome of mucus, I conducted an overrepresentation analysis of the 51 proteins that were significantly affected by nickel treatment using the consensus pathway database. This network diagram highlights the biological pathways and processes that were significantly affected. The circles or the nodes that are brighter red are pathways that are more significant, whereas the size of the circles indicates the number of proteins that belong to that pathway. The nodes are connected with lines known as edges, and the thicker the line, the greater the percentage of genes that they share, whereas the color of the edges indicates the number of proteins from the input list shared between the nodes. Although there are a lot of pathways to see here, I will only discuss a few of interest, namely the neuron projection development pathway, interleukin 12 signaling, and RAS and RAF signaling. Similarly, the 1240 proteins that were significantly affected by nickel treatment in plasma were analyzed for overrepresentation of pathways. Here, I again will highlight some pathways that are of interest and similar to the ones identified in mucus, namely interleukin signaling, neuron projection development, and RhoGDPA and RAS signaling. Interleukin pathways were affected in both mucus and plasma. The plots on top show the dose-dependent response of some selected proteins on different days in mucus, and the ones on bottom show the dose-dependent response of selected proteins from plasma that were implicated in the interleukin pathways. Although similar proteins were not shared between the two, proteins related to the same pathway were identified. Interleukin 12 is a type of cytokine and was identified in mucus. The interleukin 12 cytokine family have been found to play a major role in skin allergy, and in humans, nickel is a well-known skin allergen. The interleukin 12B is a subunit of the cytokine interleukin 12, which is expressed by activated macrophages. It functions in both innate and adaptive immunity. It associates with interleukin 23 to activate the JAK-STAT signaling cascade and promotes the production of pro-inflammatory cytokines. Although interleukins were not identified in the blood plasma, proteins such as JAK1, SOX3, and STAT5 exhibited a dose-dependent response and are involved in several interleukin signaling pathways. This could suggest that nickel induces an inflammatory response in fish, similar to the one seen in mammals. Proteins involved in RhoGDPase, RAS, and RAF signaling were also impacted. Rho family of GDPases belongs to the RAS superfamily of low molecular weight guanine nucleotide binding proteins. Some proteins of interest and the ones that commonly occurred in these pathways are highlighted in the figures on the left for mucus and on the right for plasma. These signaling pathways play an essential role in regulating neuronal morphology such as dendritic arborization, spine morphogenesis, growth, cone development, and axon guidance. Therefore, it can be inferred that the proteins involved in processes like neuron projection and axon guidance were also significantly regulated in response to nickel as seen in the previous slide. While Rho activation often elicits neuronal death, RAS signaling can promote neuronal survival and proper functioning of neurons. The GTP-bound form of active RAS leads to the activation of its direct downstream effector RAF, which encodes a serine threonine kinase and activates the MAP kinase protein. As discussed in the previous slide, neuronal morphology like neuron projection and axon guidance is altered by RhoGDPA signaling pathways. A study on mice exposed to 10 milligrams per liter of nickel in drinking water for 12 weeks revealed that nickel exposure caused learning and memory impairment, along with reduced dendrite complexity in their hippocampi. There were many proteins in the study for mucus and plasma that were identified to play a role in these pathways, but I will only focus on a family of proteins specifically known as semaphorins that are known to serve as axon guidance ligands. Semaphorins are also implicated to play a role in immune cell migration and regulation. This was observed in a study with mice keratinocyte cell lines where the deletion of SEMA3A reduced nickel allergy and regulated the macrophage polarization toward an anti-inflammatory direction. Here we can see the decrease in abundance of SEMA3E and increase in abundance of SEMA4D with increasing nickel concentrations, while SEMA3E and SEMA3B abundances remain fairly constant. The difference in expression of isoforms of SEMA3s in fish could be due to species-specific differences in response to nickel allergy. These observations suggest that the immune system and nervous system are closely interlinked and can be controlled by signaling pathways such as RhoGDPases. To conclude, the current study presents novel findings of the response of fish to nickel exposure, which were documented in other animals but not in fish. The proteome of rainbow trout is also seen to be sensitive to nickel exposure at low doses. Interleukin pathways are affected, indicating activation of immune system by cytokines at the epidermis. This is also reflected in the plasma proteome. Additionally, neuronal processes were also impacted, as seen in both mucus and plasma proteome. These are likely due to the alterations in abundance of proteins involved in signaling by RhoGDPases and RAS signaling. Neuronal and immune systems seem to be interrelated since they share many proteins, and responses in the study do not necessarily indicate an adverse outcome of toxicity, but may provide information about early signs of molecular initiating events that lead to adverse outcomes. For future directions, toxicology studies should study impacts of nickel at lower levels for longer durations of time. As seen in this study here, time can have a stronger impact on protein abundance than nickel treatment when examining impacts at lower doses. Additionally, multi-omic studies can provide a better understanding of the processes regulated at the molecular level, which can be used to establish mechanisms of toxicity. And finally, I would like to thank my amazing lab members, especially Dr. Danina Simmons, who's been an amazing mentor, and Alice for guiding me with statistical analysis. I would also like to thank the audience for listening to my presentation.